So thanks, Alex, for the invitation. It's wonderful to be among friends and colleagues to share some of the work that we're doing in my lab. So the purpose of my lab is to have fun, right? So I purposefully started out not wanting to be a professor. My dad was a professor. I told him I never want to do what you want to do, and I've been a failure in life, right? So that's the thing. So what we want to do is to do sort of interesting things, novel things that bring joy. That's the whole idea. My research is sort of split into three areas. One is in sensors, another is in medical devices. So we make these oxygenators for neonatal babies. And then the third, which has grown over the past 10 years or so, has been in the area of tissue engineering and cultivated meat. So I'll go a little bit into sort of what everybody else is doing, but it's more of a personal journey of how I or my groups got started in this area, how we stumbled, what we realized, and then where we are at this moment, right? So that's the goal. And I'd like to thank all the funding agencies that funded us to have fun. So that's really important. Um, all right. So what are the uses of tissue engineering? Broadly, there are three sort of main applications of this technology. The first one is in the area of drug discovery. So you want to create these tissue mimics that just so recapitulates the biological phenomena that is happening in our body so that you can test numerous drug candidates on them, identify which of these candidates are promising, move them forward to animal trials, and these days, there are some possibilities that it can move directly to human trials, right? So what you really want to do in the first aim is to sort of create tissue mimics, not exact tissues, but something that recapitulates some of the fundamental phenomena that you're interested in for that particular disease. Another is in terms of in vivo bioreactors. And there, what you want to do is to create structures that can recapitulate some of the function of the tissues. So for instance, producing hormones that are of interest that the body has stopped producing, for instance, and you can implant these into the body and then potentially connect them to circulatory system, and they will sort of produce this additive phenomena of making say, some of the hormones, in this case, for example, liver-related hormones. And then finally, the holy grail is, can you recreate an entire organ in all its complexity, different cell types, integrated vasculature, so that if organs get old, you can take old organs off, put new organs in, and so on, right? So these are sort of the broad application areas, and progress has been at different paces in these different segments, right? Additive manufacturing is a very old area. So it got started in the 70s and the 80s. And broadly, there are like 7 different classifications of additive manufacturing. So it starts with material jetting. You can think of it more like an inkjet printer, printing material out. Binder jetting is a similar kind of things for materials that do not polymerize. Then you have vat polymerization, which is something that let me take a laser pointer. Okay. So vat polymerization is more similar to a stereolithography system. This is one of the very early additive manufacturing processes that got started. And then material extrusion, which is quite popular and everyone knows about it. And then there are things like directed energy deposition using a laser to deposit, melt materials. Same thing, powder bed fusion is also a similar process. And then another old technique called sheet lamination, right? So all of these in some form or the other got started in the 80s. And the driving force behind them was this new technology at that time, which was photolithography. So photolithography is basically you make 2-dimensional masks and create patterns onto flat surfaces, and then you slowly try to build it up. And if you see many of the 3D manufacturing processes that you have these days, what they do is they take a 3-dimensional object, segment it into 2-dimensional slices, with all its complexity, try to make it, either print it, deposit, whatever means there is among these different techniques, and then build layer by layer, right? And that's sort of the essence of additive manufacturing processes. Now, these have been around. They have grown to become mainstream processes for plastics, for sure, increasingly so for metals and ceramics. But living matter is a different ballgame, right? So you can't use some of these things. So for example, you can't shine a high-powered laser at a cell. You're going to kill it, right? You can't heat it up. You're going to kill it as well. So the conditions that you can do are going to be benign for biological materials, and therefore only a few of them are suitable. So most of the explorations in additive manufacturing and its applications to tissue engineering has been in material extrusion of various forms, as well as vat polymerization or stereolithography methodologies, right? And an example of that would be something like this. So this is a bioprinter called Novogen MMX, developed by a company called Organovo. Not sure if they are still in business, but that was 10 years ago. And this was after about 20 years of development by Gabor Fogas. And the idea here is to do extrusion printing, extrusion of biomaterials, along with a sacrificial ink that can be then later removed away, right? So basically you print it sacrificial ink, and then you lay down your what is called structural material. And then once the process is done, you can remove it away. The innovation in their latest version was to print using not cells, but actually cellular aggregates. And cellular aggregates then tend to fuse with each other and form thicker structures such as this, which could be used as blood vessels that you can then transplant into patients and other things, right? Others during that period of time have also investigated use of other micro manufacturing processes, such as casting and molding techniques. One of the challenges in any of the tissue engineering processes is that as you go thicker, you need to perfuse nutrients, remove waste from these cells, and therefore any tissue that is greater than, say, 500 micrometers in thickness will become necrotic because of lack of oxygen, lack of nutrients, and ineffective removal of waste products. So you need to have a vascular network, or at least a perfusion network. And some of the first attempts to create perfusion networks was to cast hydrogels loaded with cells onto microscale molds that then mimic these perfusion networks, and then take them off and then layer by layer stack them up in order to create a three-dimensional structure that can be completely perfused, right? And so various gels have been used, such as agarose, gelatin, and collagen, acrylate gels, and so on. The other approach is to use sacrificial materials. So you can build a scaffold of sacrificial materials. In this case, this is an example of sugar being 3D printed into a scaffold-like structure, or gelatin being 3D printed into a scaffold-like structure. And then once that scaffold is built, you can pour or cast your hydrogel with the cells loaded in it into that structure and then dissolve away that sacrificial material. So once again, these sacrificial materials that you use cannot be anything. They have to be non-toxic to the cells. And so only certain classes like gelatin and sugar have been used so far. So this was sort of the state of the affair about 10 years ago, right? And then we thought, well, this is a ripe area to sort of get into and have fun. And the reason was because additive manufacturing tools were becoming less expensive. So the tool that I showed you from Organovo costs about $500,000 or so. That was their sale price. But around the same time, you could get this open source printer. This is called a RepRap printer. And it was $600 at that point of time. And it's an extrusion printer. So it has a print head that heats up a polymer and extrudes it in order to form three-dimensional structure. The nice thing with this kind of a printer is that not only is it cheap, but it's also, the source code is open source. So you can hack into the code, you can modify it, you can do whatever you want, right? And because we didn't have any funding for this area at that time, this was a good way to get started. But it's an interesting problem to have because a lot of people around the world will have similar kinds of problems, and therefore bringing the cost of entry down is a viable way to expand opportunities and interests and research in this particular area. So we took a printer like this, took off the thermoplastic print head, and then introduced our own microfluidic print head. And it's a very simple print head where you have like an L-shaped channel, and we had a groove through which you can insert a needle right at the center of the channel. So you have a coaxial setup that you can manufacture very easily using standard parts that you can get. And what we started out was to flow a cross-linking agent at the center, and a cross-linkable polymer on the outside. And so our example was calcium alginate. So you flow alginate on the outside, calcium on the inside, right? And when you do that, the calcium immediately starts cross-linking your alginate inside out, right? And that was sort of the difference there. And so your walls inside become stronger. And as the calcium diffuses out, you get more and more of the cross-linking. But the polymer outside is still a polymer, ungelled polymer, and it is capable of flow. So typical approach that people do is to take this kind of a setup, either a solid core or a hollow core kind of tubular nozzle, and then put it into a calcium chloride bath, so that not only can you polymerize the inside, but you can polymerize the outside, and what you get is a tube, right? So this kind of work has been done. There's a company in BC called Aspect Biosystems who have popularized this method, combined it with 3D printers in order to get three-dimensional structures with it. So what Rana, my PhD student at that time, did was, instead of doing it into a calcium chloride bath, she printed directly onto the substrate, the print bed of the 3D printer itself, right? And when she did it, what happened was that the insides cross-linked, but the outside hadn't cross-linked at the time when it came out of the nozzle, and therefore the gel actually spread out and became a big mess, right? But what she did cleverly was to then create a pattern, right? And that's what you can do with a 3D printer, right? So you can create any patterns that you want. And in this case, she created like a zigzag, or what is classically called as a log pile pattern, right? You go up and down and you write it. And if you space it correctly, what ends up happening is that the gel that is spreading, or the alginate that is spreading from one line, overlaps with the next line, with the next line, with the next line, and soon what you get is a slab of gel with a single channel running back and forth through it. So you have a single inlet and a single outlet, completely perfusible slab of gel. And if you can put anything into the gel, you can put different extracellular matrices, different types of cells, and so on and so forth, right? And so the attractive feature in this is non-biomimetic, right? So nowhere do you have a log pile pattern for perfusion network in biological systems. But nevertheless, it is very useful because it is very easy to connect to a vascular network, right? So a surgeon, and they open up a patient, is going to have access to a artery and a vein. And they have to have some way of connecting that to a single inlet and a single outlet. And this kind of a system is useful for those purposes. So she demonstrated that if you can do something like this, you can get these hollow gel layers with hollow channels embedded in it. You can perfuse them. This is perfusion of these channels with some fluorescent nanoparticles, so they don't go into the gel itself. But if you have small molecules, they diffuse well into the gel. And the cross-linking is reasonably fast enough that you can go one layer up and down and another layer back and forth, right? And it remains separated, right? So these channels don't fuse with each other. They remain separated, and therefore you can create multi-layer structures, right? So within about 10 minutes or so, you can print up to 10 layers, a really dense or thick tissue with, again, a single inlet and a single outlet. And what we also went on to show was that these kinds of systems can be perfused, and because of that perfusion, cells in them live and are viable over long durations of time, while same thickness bulk gels, which don't have these channels, the cells in the interior part of it actually die off. Then she thought, well, I can go fancier, and not just can I do one coaxial setup, but I can scale it up, right? And that's the beauty of this kind of a system, is that you can have not just one, but you can have two, three, as many as you want. And because the flow in these channels is very low in Reynolds number, you have purely laminar flow, and the layers don't mix with each other, right? So you can segment and radially pattern whatever material that comes out of this nozzle, right? So this is examples where you can have a hollow core, and in this case, she dyed the gel with a blue dye, and then in here she dyed the gel with a yellow dye. And what you can see is that those structures or those materials stay in those locations, even when they are printed onto a flat dry substrate. And she demonstrated that, okay, not only can I do two, I can do three layers very easily. And when you sort of draw lines adjacent to each other, there's overlap, and the final layer actually overlaps with each other, but you can create this kind of a radial geometrical arrangement. What you can do with that radial geometrical arrangement is to introduce different materials in different layers, right? So for instance, what you can do is closer to the perfusion channel, you can introduce a gel which has endothelial cells in it. And so they can migrate out and they can form the blood vessels. And then farther away from it, you can have fibroblasts, which can then form the structural components of the tissue itself, right? And what you can see here is that the red cells are located here and that the green cells are located all through the construct itself. You can also use this radial geometry or segmentation in order to introduce different kinds of extracellular matrices. So alginate is not really a good matrix for cells to propagate and grow and form tissues. On the other hand, materials like collagen and fibrin are much better. And therefore, what you can have is introduce alginate on the interior just for structural strength. And then on the outside, you can introduce a mix of alginate and collagen for the cells to actually connect with the extracellular matrices and form tissue-like structures, right? So that was what she had did by the time she completed her PhD. And what we found was that, well, radial patterning is great. It's useful in some applications, especially if you want to create some vascular networks and so on. However, what you want is a complete ability to fabricate or impose patterns onto these structures in all 3 dimensions, right? So you need to have greater flexibility rather than just radial arrangement. And so the next student, Lereza, he used this technique called Excel. It's a hybrid manufacturing technique. It's a combination of additive and subtractive manufacturing in order to create 3-dimensional geometries as desired. And it starts with a scaffold. So in this case, the scaffold that we had was paper, but you take any porous scaffold that you have. And then what you do is you then immobilize your cross-linker onto the scaffold, right? So for example, if your cross-linker is calcium, you soak the paper in calcium, dry it, so you have calcium embedded onto your porous matrix. Then next, what you do is you come in with a 3D printer and you can print any pattern with the gel, the cross-linkable polymer material, right? In this case, alginate, but you can have different combinations. You can use a pH modifier for your cross-linker and collagen in order to do collagen cross-linking, or you can do fibrin in a similar kind of way as well. But the idea here is that as this polymer touches onto the substrate, it wets the substrate. Once it wets the substrate, the cross-linker goes into the polymer, cross-links, and prevents the spread of the gel. Otherwise, the porous matrix is going to allow the gel to spread very, very quickly through capillary action. And by this way, you're arresting that spread and retaining the gel pattern onto your porous matrix, right? And then what we also did was, if you look at a tissue structure, you also have hollow spaces, like the vascular regions or the perfusion regions are hollow spaces through which you want to perfuse extra or other liquids into these things. So in order to do that, what we did was we used this process called xerography, which is a glorified name for paper cutting, right? And so you have a plotter with a blade on top of it, and you can cut any pattern that you want, and you can use it to cut the porous matrix underneath it. Right? So these are very thin paper-like matrices. You can cut whatever pattern that you want, and usually what you try to do is to cut the pattern that is similar to the network or the vascular network that you are interested in. And then after you do this, then you can come in with a printer and print different cells in different regions, right? So gels with different cells, so endothelial cells in the inside, and then fibroblasts or muscle cells on the outside, and it's up to you to design what patterns you're interested in. So we have created patterns like this where you can co-locate one cell type with another cell type, and you can create very, very sharp boundaries in this case. And these are very useful to see how one cell type interacts with the other cell types in a 3-dimensional geometry, right? And whether there are migrations from one region to the other. You can also create these what is called as migration assays. These are where you print the cells in a certain pattern, and then you can feed nutrients to it or drug candidates which will promote or affect migration, and this is important in wound healing and other kinds of applications. So the idea here is to create different kinds of geometry. And finally, what Alireza also did was to show that we can actually create perfusible networks. So he did 3 layers. The first layer had HUX on the inside, which is endothelial cells, and then fibroblast cells on the outside. And then the top layer had, again, a similar pattern, except it didn't have that cavity or the hole. And then what you can do is to perfuse, and that perfusion can put the fluids into these channels and take it out and create three-dimensional networks in this particular way, right? So that's sort of the second work. And then what we figured was that, well, this is all fine and great. We can get all of these things. But so far, we've been only working with alginate. And alginate is not present in our body. So we wanted to work with systems where you are working with extracellular matrices that are present in the body. And collagen by far is the most abundant extracellular matrix that is present. So we looked at collagen-based systems and how to create macro scale shapes in these systems. And one of the techniques that we came up with was this self-assembled constructs. So collagen, as you change the pH to more basic, excludes water and self-assembles itself into sort of a triple helical structure, and that then creates a much more solid construct, and which you can handle very easily. And so what we did was to create microscale molds using microfabrication approaches of different shapes. So for instance, this is sort of a dumbbell shape, a cross, a square, or a rectangle. And if you seed it and change the pH, what you end up happening is to recreate these cellular assemblies in the same shapes as the mold. So you can create any shape of the mold with microfabrication, and you can impose on it. Not only can you do that, what you can also do is you can segment this overall shape and seed collagen-based bioinks loaded with cells, different types of cells in different regions. And then as they assemble, they sort of merge with each other and form a single monolith, right? And you can, within 6 hours or so, you can pick it up with a tweezer. You can do whatever you want. It functions like a tissue by itself, right? And so that was very interesting. We showed some of the fundamental aspects. So the more the cells that you have, the greater the shrinkage is going to be. So initial assembly is because of collagen forming sort of these fibrous structures, and then subsequently the cells attach to the collagen and impose traction forces. These traction forces exclude more of the water from that matrix, and then it leads to further shrinkage itself. We also demonstrated that using this, we can load different amount of cells and different amount of extracellular matrices in it. And this is important because if you're creating a model where you're studying cell matrix interactions, and that is the key question that you're going to design a drug candidate for, then you can go with low cellular loading, where the cells are going to interact with matrix mostly. Or if you go to high cellular loading, then you can get in vivo-like cellular densities, right? And where the questions that you ask will be about how cells and cells interact with each other, and are they synergistic, and how they interact with the drug candidates as well. But this tunability is the key, and it's a very simple process of just initial composition change, and that will give you the right kind of matrix and composition in order to address the questions that you're interested in. We also found that different cells also have different amount of shrinkages. So cancer cells don't shrink as much, while other cells, endothelial cells and fibroblast cells actually shrink quite well, right? And so this is an example of segmental seeding. So in this case, we have inserts that go into these different regions and prevent a fluid that is deposited here from flowing back. And once you do that, you can do sequential seeding over 30 minutes or so. And because you have that initial assembly, after 30 minutes, it's not going to mix with each other, and you get that segmentation very clearly. Now, this kind of an interface is particularly important in biological systems, right? So for example, if you look at our muscles, right? The fat and the muscle cells or structures are not homogeneously mixed in these things. So you have your muscles as in the fiber form, and then you have fat in the form of sort of these globular structures surrounding the muscle itself, right? And so the question that we wanted to ask was, is this structure, the macro structure, really important, right? Does it make a change? And we did some experiments where we are doing co-culture. So we had two wells that were connected to each other, but we can segment them while seeding and start forming the collagen structures initially and then merge them together, right? And so we created constructs where we had sort of fat cells or pre-adipocytes on one side, muscle cells on the other side. And so this is a heterogeneous distribution of cells similar to what you find in vivo. And then we also mixed them together, seeded the same number of cells in the wells, and that's a homogeneous distribution of cells, right? And then we tested the response of these things to various things. So what is the metabolic activity, the lipid content, the protein content of these things, and their response in terms of fatty acid release to some potential drug candidates. And what we found was that the fat muscle heterogeneous co-culture versus the homogeneous co-culture was actually significantly different from each other in the fatty acid release. So it is really important that we do maintain the structural configuration that we see in vivo. Otherwise, the response to these drug candidates are going to be quite different from each other. And that sort of emphasizes the kind of macro structure creation that additive manufacturing can bring forth to this area. So this was all great, but we can only form solid structures at this point of time. And so a new student, Aydin, came in and he said, well, I want to make hollow structures that I can perfuse, right? And so one of the things that he tried was to create molds like this, where you can insert a wire, and this is a metal wire coated with Teflon, into sort of the central portion of the well, and then you can put the collagen bioink with cells in it, and what happens is as they assemble, they form around the wire. But because you have Teflon coated wires, the cells or the extracellular matrices don't attach to the cells, to the wire itself, and you can remove the wire, creating a perfusible kind of construct. And what he has shown is that he can create complex perfusion networks, right? So in this case, this is where you have a single inlet and two outlets. This is a crisscross geometry, and you can create very large structures. These are about 7 centimeters by 7 centimeter structures, all made of endothelial cells with collagen gel in it. The nice thing with these things is that he had, in fact, solved one of the key problems in any tissue engineered product, which is how do you interconnect with the product itself. So you can form nice cellular assemblies and so on. Often the challenge is to form good seals when you connect like a plastic tubing with these materials. And by using this self-assembly process, what he has done is, so these are plastic connectors that you can buy at McMaster-Carr, for instance. And what you see is that because it is self-assembling and shrinking, it imposes a compressive force on the plastic part and creates a tight seal that we can actually apply pressure and perfuse it, and without, I mean, it won't go to a very high pressure, but nevertheless, we can apply a pressure to perfuse and it won't leak, right? And so this assembly is very nice in terms of interconnection to plastic devices and creating a very tight seal for transport related studies. What he also then showed was that because the assembly process is fast, within a few hours or so, you can sequence the way that you're introducing the cells. So you can first initially load with one kind of cells and then come back after, say, a few hours, load with another kind of cell. And you can get these, again, core shell type of assembly or radial patterning, right? So in this case, you have a central hollow core, and then you have one type of cell around it and another type of cell assembled in this fashion. You can also create an axial assembly, again, using inserts. You can introduce different cell types, all with collagen matrix. And then when you take off these inserts, then they sort of fuse with each other and form a monolithic structure with a single channel perfusing through that. And these are quite important because you can do cancer studies with this. So you can include cancer cells at the center and then identify how these cancer cells actually spread to the neighboring tissues and all of these things. What he also found when he seeded endothelial cells in it is that the cells, as you culture them, actually initially they are homogeneous. So this is day one, where the cells are loaded into the bulk of the matrix. But over time, they actually propagate to the outside and form a very thin membrane, right? And so endothelial cells form endothelial membranes, which is sort of their natural thing to do. And these membranes form very tight junctions. So this is a staining of a surface receptor protein, indicative of tight junctions forming between these cells. And the entire surface of this construct is one contiguous endothelial layer. And what is shown through permeability studies from inside out is that this format is very good for transport studies across membranes. And this kind of permeability is very close to in vivo permeability and one of the best that you can get for models. And again, it goes back to the interconnects and the ability to seal to plastic parts that becomes key and important in achieving very low permeability in these kinds of structures. But it also points us out to an interesting thing. One is that you don't need to actually recreate the exact three-dimensional geometry of cells in the right locations and the extracellular matrix in the right locations in order to form a functional tissue, right? And as a mechanical engineer, 10 years ago, that was my thought, right? So I said, well, I'll take this tissue, segment it into two-dimensional layers, put the right cells in the right location, and lo and behold, I get a tissue, right? But that's not needed because these are not inanimate objects. These are actually living things. They have, in some senses, an intelligence of their own. They respond to cues that come from the environment, to the matrix that they are present in, to the neighboring cells that they interact with, to the chemical agents that these cells are secreting, and all of these things. So as a good cook, what you should try to do is to create the right recipe. And once you have that initial conditions right, you can let the system evolve, and the system will evolve on its own with the right configuration that you require, right? So what Aiden is continuing to do is to look at using this for other barrier functions, right? So one of the things that he's looking at, I didn't have much time to include results here, was the placental membrane, and to look at how these then evolve when you seed them uniformly into the construct. Different from endothelial cells, the placental cells, when you seed them, actually do migrate, but don't form a very thin layer of 1 or 2 cells. They actually form a thicker layer of 5 to 8 cells in thickness. And the top layer of the cells are fused with each other, similar to the syncytiotropoblast of the placenta. And the cells in the bottom are similar to the cytotropoblast layer, where they are not fused with each other, but they serve as a feeder layer to the cells above, right? And so that again sort of is very promising because you don't need to define exact geometry and structure at the micro scale. You can let the system evolve into the state it wants to be in if you give the right cues associated with it. So apart from the hollow tubes, we also wanted to make larger tissue-like constructs. One of the techniques that we came up with was a very low-cost method. Take a tube, typically an oxygen permeable tube like silicone, insert 2 pins into this tube, and put in our collagen ink, collagen with cells mixed in it, and they automatically anchor onto these pins and form tube-like structures. These are solid core tube-like structures. And because these pins serve as anchoring locations, they exert a mechanical stimuli, stretch onto these tissues that are forming. And because they are conductive, you can apply an electric field in order to impose biophysical stimuli onto them. We've shown that we can do a variety of things. We can form long, short tissue constructs in this way, and we've made tissues or tubular type tissues of more than 4 or 5 centimeters or so in length very easily. The interesting bit is that if you take some of these things, they form within, say, 5 to 6 hours or so. If you take them and put it into a well or a petri dish, then the cells are not oriented in any particular way. They're sort of facing in every direction. If you retain them in the tube as you culture them over 3 or 4 days or so, there is some degree of orientation. But if you apply an electric field, then there is a greater degree of orientation and fusion with some of these cells, right? And therefore, this shows some importance of creating biophysical stimuli, which is generally tend to be ignored in tissue culture area, as a way to stimulate the cells in order to achieve the kind of organization that you really want in these structures. And so these are examples of something where we have created like 10 centimeter long long tissue constructs, one single monolithic tissue. And depending on the mold that you have in the form of tubes, you can basically have different shapes along the axis of these things. Again, remember, these are all connectors that we buy and connect them together. So we have actually pulled this tissue construct out of it without tearing it apart. So that shows the robustness of this kind of an assembly process. The final technique that we came up with was to make sheets. And here what we wanted to do was to also not use collagen as an extracellular matrix, deal only with cells and have the cells produce their own extracellular matrix, right? So it's a much, much simpler system, gives you better properties associated with that. So, so the way that we did this was to culture the cells in well plates and till they achieve confluency, so they are touching each other, and then introduce a pH switch. So we move this in media into a slightly acidic media for about 30 seconds, 30 to 45 seconds or so, and then quickly we switch it back to a basic media for another 30 to 45 seconds, and then go back to neutral media. All right. This process, the acidic switch, actually helps delaminate the sheet. And when it delaminates, it sort of connects with the neighboring cells and comes off as a single sheet. If you overtreat it, you either kill the cells or you lead to this tissue aggregate like structures, like a crumpled paper kind of structure that you get. But if you quickly switch to the basic media, the sheet-like assembly remains, right? And you can pick it up with a tweezer, you can do whatever you want. In fact, you can stack them up one on top of each other and they fuse to form three-dimensional structure, right? So this is a technique very similar to this layer by layer lamination technique that I talked about earlier, right? And that was the original additive manufacturing technique. You create any patterns that you want in 2 dimensions, stack them up, and you can create a 3-dimensional structure. And that's what we wanted to do. With this case, we characterize this assembly process. Again, it's a very fast process, typically 6 to 8 hours or so, and you can get very nice assemblies in this way. The shrinkage of a single layer of cells is quite high, more than 50% or so. But if you stack multiple cells together, then the traction forces that the cell exerts is balanced by adhesion between the 2 different layers, and then the shrinkage is lesser. So similarly, if you apply this onto a pre-existing tissue, also you can restrict the shrinkage of these sheets. We've shown that these sheets are about 50 micrometers in thickness, about 5 to 8 cell layers in thickness once that initial shrinkage has happened. And you can stack multiple sheets of different types of cells to form these layered assembly. And a lot of our human tissues are layered, like brain, for example, the cortical structure is layers of white matter neurons, and so on. Similarly, kidneys are layered, lungs are layered, and so on. So this is a nice technique to form sheets and layer them to create that kind of cellular interactions, and also at very high densities. The kind of cellular densities that you can get are very in vivo-like. So using this, at that time, cultivated meat was very popular. So we said, well, let's demonstrate this. For cultivated meat, we co-cultured some of the muscle cells with pre-adipocytes, and then we differentiated them to show that the muscle cells form myofibers. They fuse with each other, forming longer fibers. And then the pre-adipocytes become adipocytes, generate these lipid globules, which are indicative of function of these adipocytes, right? So these globules of fat are what gives meat its taste, and the fibers are what it gives the texture. And in meat, these two are sort of the more important properties. What we showed at that time for the first time was that we can tune the fat muscle content, right? So just like you can get skim milk, 2%, 3%, 10% milk, now you have a possibility of getting meat, which is very predefined in terms of the fat protein content of it. And we were able to assemble that into multiple sheets. So 18 layers stacked together looks like this. And when in a dried form, it looks something like this, right? So that was the origin of this. And since then, we've been able to develop this into a large Genome Canada project involving 5 universities, about 15 investigators or so, looking at the entire ecosystem of cultivated meat, right from the cells themselves, the growth media, bioreactors, scaffolds, tissue engineering of meat, including optimization of all of these processes, and then looking at sort of some of the consumer preferences, ethical, social, cultural risks associated with it, as well as life cycle analysis on to whether it provides environmental benefits or not. So this is a newly started project that is ongoing. We also use the same technique for biomedical applications. So one of the immediate thoughts that we had was to look at skin, because skin is a layered tissue, has three layers: epidermis on the top, dermis in the middle, which consists of some vasculature in it, as well as fibroblasts, and then hypodermis in the bottom, which is mostly fat cells. So one of the collaborators here in Hamilton Health Sciences, he extracts mesenchymal stem cells from burn patients, and so he is able to expand these cells, and we get those cells, quickly assemble them within, say, 3 to 4 hours or so into sheet-like structures, and these sheets look beautiful. So this is the first time we've demonstrated that stem cells actually form sheet-like structures. The cells are alive, very few dead cells in them, and we can form different constructs. So we can combine these stem cells with fibroblasts to form sort of the dermis layer, and we can have individual stem cells forming the epidermis layer. We can stack them together to form the sheet-like cellular assembly that is representative of a skin precursor. We have also shown that we can make large tissue. This is a dated picture. Here it shows about 4 centimeters or so in size, but we can now go to 5 centimeters diameter easily. So these are 50 micrometer thick layers, but 5 centimeters in diameter. And you can sort of lift them up with tweezers. They don't tear, and you can handle them very, very easily. We then implanted them onto a pig burn model. So this is a full thickness burn model of a pig, where the entire 3 layers are burnt away, exposing the vasculature. And then you can sort of put this on top of it and look at the healing of the wound over 40 days or so. And what we find is that our cell sheets integrate quite well. They vascularize well. They have very little scar tissue inbuilt in it, and therefore these are suitable ways of creating skin-like substitutes for burn patients, where, especially if they're extensively burned, there's no other alternative for something like this, right? So this is, again, an ongoing kind of project, but we're interested in looking at collaborations with other groups that are looking at sort of barrier membranes or sheet-like membranes who have these cell sources and who want to assemble them into larger tissue-like constructs for regenerative medicine applications. The microstructure of these sheets look very nice. So as time progresses, we see more and more tighter integration of the cells with each other in the sheets. And as you culture them more and more, they become thicker because of that shrinkage if they are left unconstrained. And as they become thicker, what you see is some kind of structural orientation evolve from this. So the surface layers, the cells are all aligned parallel to the surface. In the interior, you have more of an isometric arrangement. They also produce a lot of their own ECM. So we don't need to add extra ECM or ECM from other animal species, which is useful in regenerative medicine. We can form various shapes and structures. So here, these are various shapes that we have created, and we have taken one of these shapes, and we can assemble them. We can stick the tips onto each other, and we can create any complex geometry that we want, right? So it's like Lego. So you have these basic bricks. You can assemble any complex 3-dimensional geometry that you want from those basic building blocks and elements. And we've shown that we can pattern one cell type onto the other. This is important in meat for marbling kind of applications. And then finally, we've also integrated vasculature. So this is where we co-culture endothelial cells, and it forms automatically microvasculatures that are perfusible in these constructs. So I think I'm running out of time, but I'd like to conclude by saying that this has been sort of a journey of learning for myself and my group. Initially, I thought like a mechanical engineer and was excited about bioprinting and so on. More and more as I get into this field, what I realize is that the simpler the system it is, the more robust it is, the better it is for regenerative medicine application and for integration into the human body. And so lately, we've been exploring more of the scaffold-free methods, just using cells, especially mesenchymal stem cells, so that we can have less immunogenic reactions and then allow the cells to generate their own extracellular matrices. That I'll conclude. I'm happy to answer questions.
So for the skin, you need stem cells in the same patient then, right?
No, so that's the interesting bit. So So there is this autologous therapy, which is where you take the patient cells, these are primary cells, and then you can create these sheets. But it takes a long time. It takes like 30 weeks or so to do that, and patient is burnt, right? So that's not ideal. But you have what are called induced mesenchymal stem cells. They are derived from pluripotent stem cells, but they don't have the same immunogenic reaction as the pluripotent stem cells. So, and my collaborator in HHS, he has this way of making that. So they lack some of the surface proteins which generate this immunogenic reaction. So you can generate them. They don't need to be patient specific. You can generate them and create these structures. And what we are hoping to do is to sort of have them ready, and then we can introduce these things. So there are some intricacies in how do you create blood vessels using those type of cells and so on, which are still unanswered, but that's the general way to go about doing it.
That cell density, does it come from proliferation, or does it come from like the seeded density itself that remains?
Yeah, so it evolves over time, right? So we have to have a minimum critical cell density. So in the paper we have identified what that minimum is in order to form that initial sheet, right? But that once you form that initial sheet, the cells exert traction forces among themselves. Then they also generate ECM and then attach to the ECM as well. And so it is a self-regulating system. The cells, so that's why you get from a monolayer to a multi-layer. So these sheets, although they are sheets, they are 5 to 8 cell layers thick. And over time, as they pull the periphery, the thickness increases if you let it unconstrained, right? And so they exert forces till they can bear and go to a state, we think, which is sort of the ideal biomechanical situation that they want to be present in, right? And so we don't exert any extra forces and so on. Unconstrained, they evolve, but they remain sheet-like structures except thicker.
How much is the TP? Reflected from tendons or from synthetic sources? There are very different types of
Yeah, so it's a bovine collagen. It's a bovine collagen that we use, and I don't know where it's not tendon, I think, but we are slowly looking into other sources of collagen as well for that work. In case of the scaffold free, we don't use any extra.
study pharmacokinetics.
It should be possible, right? So we can create, I mean, all of these structures we can create in different formats. And the advantage with these systems is that one, you are using sort of more native materials and cells. The other is that if you require perfusion, right, then that is something where we can assay quite easily. So it is feasible. We haven't done that yet. We mainly focus on the biofabrication, not necessarily on the applications. But yeah, happy to collaborate for applications. Yeah.
Ravi, how long do cells survive? You showed some data where you showed 8-day or 7-day images. But when you put these cells into either the collagen or on the paper or in the various constructs, what's the sort of survival of the cell?
I mean, as long as they have access to nutrients, they survive well. So maximum that we've done is like 14 days kind of thing. And after that, the student gives up, right? I don't need more data. That's enough, right? So I think they're okay. And with the implanted stuff, we've gone, so those cell sheets that were implanted onto pigs, we've looked at over 40 days, right? And they've survived well.
What scale do you take these things on? Because these look like the pictures that you were showing with small bits, but can these actually be scaled up to?
Yeah, so that's the challenge, right? So in the sense that we need enough cell mass to do this. So the project, so that was sort of the genesis of why we got funded, but it is not the only approach that we're looking at. So we're looking at the entire value chain, and part of the tissue engineering bit is that how can we sort of combine plant protein with animal protein so that you get the right texture and taste, but you can also create nutritional value in it, right? So essentially, if you can't distinguish that it is not 100% animal protein and you blend in plant protein in it, so it's healthier to eat as well, but it's also cheaper to make it that way. So that's one of the things that we're trying to do is to create that blend so that we have some cells in it, but making the entire structure out of cells is right now quite an expensive process. I think companies are trying to do this, but most of the companies are looking into this hybrid meat where they have a lot of plant protein and a little bit of animal protein in order to get that texture and taste.